morning. Our today's topic is oxidative decarboxylation of pyruvate. The pyruvate or the pyruvic acid is a production of glucose via glycolysis. Every foodstuff like protein, fat, and carbohydrates, protein after metabolism of amino acid and fatty acids after metabolism for the beta oxidation converts ultimately in acetyl coenzyme A. The acetyl coenzyme A is a universal pathway for the all foodstuff to enter in a oxidative energy production system, that is citric acid cycle, where it produces NADH2 and FADH2. Those goes through the respiratory electron transport chain and produce energy. Here we will discuss the pyruvate and acetyl coenzyme A. Acetyl coenzyme A converts in carbon dioxide and other FADH2, NADH2. Conversion of pyruvate into acetyl coenzyme A is a very complex process in which three enzymes E1, E2, and E3 and five coenzymes assisted this process to complete, and this process is known as oxidative decarboxylation. The oxidative decarboxylation is essential to convert pyruvic acid to acetyl coenzyme A. In aerobic organisms, glucose and other sugars, fatty acids, and most amino acids are ultimately oxidized to carbon dioxide and H2O via the citric acid cycle and the respiratory chain. Before entering the citric acid chain, citric acid cycle, the carboskeleton of sugars and fatty acids are degraded to the acetyl group of acetyl coenzyme A. The form in which the cycle accepts most of its fuel inputs. Many amino acid carbons also enter the cycle this way. Although several amino acids are degraded to other cycle intermediates, here we focus on how pyruvate derives from glucose and other sugars by glycolysis, is oxidized to acetyl coenzyme A and carbon dioxide by the pyruvate dehydrogenase complex. Pyruvate dehydrogenase complex or PDH is a cluster of enzymes. Multiple copies of each three enzymes are located in the mitochondria of eukaryotic cells and the cytosol of prokaryotes. Here we can see the overall reaction of oxidative decarboxylation of pyruvate to acetyl coenzyme A by the PDH complex. PDH complex, pyruvate dehydrogenase complex, comprises three enzymes, namely first decarboxylation of Pyruvate produces hydroxylethyl thymine pyrophosphate and then the oxidation of the hydroxyl group to an acetyl group. The electrons from this oxidation reduces the disulfide of lipoid bound to E2 and the acetyl group is transferred into thioester linkage with the thio group of reduced lipoid. The name of all three enzymes is even dihydroxy dihydrolipoid transacetylase. This is E2, uh, pyruvate dehydrogenase E1, and dihydrolipoid N hydrogenase is E3. The pyruvate dehydrogenase complex requires five coenzymes. The combined dehydrogenation and decarboxylation of pyruvate to the acetyl group of acetyl coenzyme A requires the sequential action of three different enzymes and five different coenzymes or prosthetic groups: thiamine pyrophosphate (TPP), flavin adenine dinucleotide (FAD), coenzyme A, nicotinamide adenine dinucleotide, and lipoid. Four different vitamins required in human nutrition are vital components of this system: the TPP or the thiamine, the riboflavin in FAD, the niacin in NAD, and pantothenic acid or pantothenate in coenzyme A. We have already described the role of FAD and NAD as electron carriers, and we have encountered the TPP as a coenzyme of pyruvate decarboxylase. Coenzyme A has a reactive thiol group that is critical to the role of coenzyme A as an acyl carrier in the number of metabolic reactions. Acyl groups are covalently linked to the thiol group, forming thioesters because of their relatively high standard free energy of hydrolysis. Thioesters have a high acyl group transfer potential and can donate their acyl groups to a variety of acceptors molecules. The acyl group attached to the coenzyme A may thus be thought of activated for group transfer. The fifth cofactor of the PDS complex required as thiol groups that can undergo reversible oxidation to the disulfide similar to that between two cysteine residues in a protein. Because of its capacity to undergo oxidation reduction reactions, lipoid can serve both as an electron hydrogen carrier and an acyl carrier as we shall see. These pyruvate dehydrogenase com- complex has three distinct enzymes. These enzymes are pyruvate dehydrogenase, dihydrolipotransacetylase, dihydrolipoyl dehydrogenase. The structures of all three are as follows. The active site of E1 has bound TPP and the E3 has bound FAT. E3 bound FAT. E2 has three functionally distinct domains: the amino acid terminal lipoyl domain containing the lipoyl lysine residues, the central E1 and E3 binding domain, and the inner or acetyl acyl transference domain. These are the structure of E1, E2, and E3. Such type of reactions also present in the Krebs cycle, where the organization of the PDH is similar to that of enzyme complexes that catalyze the oxidation of alpha ketoglutarate and the branched chain alpha keto acid. Thus, we can say that the process of oxidative decarboxylation is a very complex, and the enzyme pyruvate dehydrogenase complex involves the different actions to complete this process. Thank you.